we are very happy to present the new data on the optimized formulation. And actually, for that question, let me maybe share some of the data we are presenting. So the vaccine formulation we're using is a liposomal vaccine, as displayed here on the in the middle of the slide. On that liposomal vaccine, we express the antigen. So that is the Aβ 1 to 15. That has been selected because there are specific B cell peptides or peptides that have epitope for B cell induction. We had an initial vaccine, and that is actually very similar between our ACI-24 amyloid vaccine and our ACI-35 tau vaccine, where we had seen antibodies produced, but we were not happy with the boosting and the amount of antibodies. Now, in preclinical experiment, and that actually has as well been presented here at the conference, we have demonstrated that when we use this initial formulation and we add T-cell help, then we can increase the amount of antibodies, but as well the boostability. So over time, we see higher and more maintained titers with adding this independent T-cell help. What we as well see in this is that the antibodies produced very nicely target Aβ 1 to 42, but it has a nice distribution over these first 15 amino acids of Aβ. It as well targets pathologic fragments by pyroGlu, and we have data targeting oligomeric Aβ in preclinical species. So that led us to the fact that we believe this new optimized formulation will be much more suitable for treating Alzheimer's disease and the pathologic fragments. The supporting evidence from the initial studies obviously was the safety and tolerability of the vaccine platform itself. So as of today, we have not seen any adverse effects that lead us to the fact that this could be a problematic development with liposomal vaccines, and we have that with tau and the Aβ. The preclinical data, as I just referenced, and as they will be presented here, just demonstrate the improved performance of the liposomal vaccine with the T-cell help, just in terms of higher antibody response, and what we as well see in the tau vaccine, a maturation towards the pathologic species. So that is what we see as the supporting evidence. Obviously, then we have the classical tox packages for the new formulation, and therefore we think now this is fit for prime time in clinical studies, and therefore we decided to now initiate this new clinical study, the ABATE study, where we are presenting the design here today. Maybe taking a step back a little bit, in the poster we as well talk about biomarkers and how biomarkers are used in clinical trials today. And we do see two main utilities. And obviously there is a lot of data, as we'll discuss here in the conference, how we now can use biomarkers of amyloid and tau to select patients. So we and others are able now to very precisely target groups of patients in the sense of their severity, disease confirmation. So we know that individuals with amyloid and tau are having the Alzheimer's disease, but we as well can say where they are in the journey. The other thing where these biomarkers now can be used is a pharmacodynamic effect. And with the recent monoclonal antibodies, we realized that some of the clinical effects were not as strong as we had hoped for, but in general, the clinical effect that had been seen can be attributed to amyloid lowering. So there was a very nice review published here by Avgerinos and colleagues, where they looked at 4 different monoclonal antibody phase 3 programs. So this is really phase 3 pivotal evidence, and they looked into how does amyloid-PET lowering correlate with cognitive effect? And what you see is for all of these programs, for ADAS-Cog, Syria, some of boxes, that the more you lower amyloid-PET, the stronger you have the clinical effect. And I think that is as well very, very nicely supported by the recent data published on donanemab. So this is important for us because we obviously use these biomarkers now. So on the one side, we use amyloid PET scan to select our individuals, both on sporadic AD and for the individuals with Down syndrome. And then we will be able to demonstrate amyloid PET lowering at a later point in the trial. What we initially do, so we initially want to know, is there immunogenicity? So is there antibody production? Is that sustainable? Is that safe? So therefore, we take escalating doses of the drug to see if we have that antibody response. Once we found a safe and immunogenic dose, we can do two things. The one thing is we initiate the Down syndrome part to then confirm if the antibody production in Down syndrome is the same, if we have the same safety profile. But we as well can take one of these smaller cohorts. So here we talk about 8 subjects per group, and take one of these smaller cohorts and expand it to about 30 subjects. And then we can look into an amyloid PET lowering pattern of the vaccine. So we can already in this very early phase understand the amount of drug produced, because that is in principle the antibody, the polyclonal response is our treatment. So we can look into the polyclonal antibody production, and then how that relates to amyloid lowering. And we can as well then see how does this similarly is seen in individuals with Down syndrome, and how do we see amyloid reduction in the population with Down syndrome. Now, we are able to do interim analysis in these trials because obviously these biomarkers are very hard markers. We can do very meaningful analysis in small sample sizes. So that allows us to do interim analysis and very early decision making for the overall program then.